Today we're going to start our animal development unit, and we're going to be looking at 3 distinct events that take place during fertilization, and we're going to look at cleavage pattern variations between different animal embryos. Fertilization, or the combining of the male and female gametes, involves a sperm cell penetrating the outer protective layer of an egg and fusing with the egg's membrane, the activation of the egg, and the fusion of the two gamete genomes. Before we talk about the penetration process, we should first look at the outer protective layers of the egg cell. Now, the diagram on the right depicts a typical mammal egg, whereas the diagram on the left represents a sea urchin egg. Starting with our egg cell on the right, the zona pellucida is a protective layer consisting of an extracellular matrix composed of 3 glycoproteins. The sea urchin egg has an outer layer that is comprised of polysaccharides that bind receptors on the head of the sperm, but the jelly also acts as a physical barrier between the sperm and the egg cell. Inside the jelly layer is a vitelline layer, which is a membrane composed of glycoproteins. Continuing with the sea urchin example, in the front portion of the head of the sperm is an area called the acrosome, and when jelly polysaccharides bind receptors on the head of the sperm, enzymes from the acrosome are released that digest the jelly polysaccharides, and this allows the sperm to penetrate the jelly layer. After this enzyme release, actin microfilaments polymerize, forming a protrusion called the acrosomal process, and this structure contains a protein called bindin. Species-specific bindin receptors on the vitelline envelope bind the bindin proteins, and the acrosomal process then penetrates the vitelline envelope and fuses with the egg plasma membrane. This membrane fusion triggers the opening of sodium channels in the egg membrane, triggering an influx of sodium ions into the cell. This increase in positively charged ions depolarizes the cell membrane of the egg, which temporarily blocks other sperm from fusing with the membrane. This fast block mechanism deters polyspermy, which is fertilization of the egg by more than one sperm. Intracellular calcium ion concentrations increase shortly after the sperm makes contact with the cell membrane. This calcium ion increase results in the fusion of cortical vesicles with the egg plasma membrane, releasing their contents into the space surrounding the egg. This space is called the perivitelline space. This raises the vitelline membrane from the cell membrane and inactivates bindin receptors on the vitelline membrane. Therefore, any additional sperm are released from the vitelline membrane and no more bind. This is known as the slow block to polyspermy. Now, this initial lifting of the vitelline envelope away from the cell membrane results in the formation of the fertilization cone, which is the first visual cue that fertilization has taken place. As the vitelline envelope continues to separate, we then see the formation of the fertilization envelope. And here's a picture taken through a microscope in a previous semester of Bio 153 lab showing the fertilization envelope around sea urchin egg cells that have been fertilized. This increased calcium concentration in the cell also acts as a second messenger signal which serves to trigger an increase in metabolic activity within the egg. In the final stage of fertilization, the haploid sperm nucleus fuses with the haploid egg nucleus to form the diploid nucleus of the zygote. Now, in mammals, including in humans, the nuclei don't actually fuse. Instead, the sperm and the egg nuclear membranes each break down prior to the formation of a new diploid nucleus, and then a new nuclear membrane forms around the 2 sets of chromosomes. The rapid cell division that takes place following fertilization is called cleavage. During cleavage, the zygote is subdivided into a larger and larger number of smaller and smaller cells, and we call these cells blastomeres. And pictured here are various cleavage stages of aptasia embryonic development. And we can see all the way from the oocyte to the blastula stage, and we'll see later even beyond, there is no increase in overall size. There's just an increase in the number of cells. Now, there's quite a bit of variation or diversity in cleavage patterns between different types of animals. In eggs with little or no yolk, what we see is holoblastic cleavage, and this is observed in invertebrates, amphibians, mammals, and others. And in eggs with large amounts of yolk, we see what is called meroblastic cleavage. At the end of the cleavage stages, the blastula forms. The blastula is a hollow ball of cells. In mammals, it's called a blastocyst, and the interior of this hollow ball is called a blastocele, and it is a fluid-filled cavity. Now, what takes place to form the blastula is that the interior cells begin to pump sodium from their cytoplasm into the spaces between the cells. This creates an osmotic gradient, which causes water to be drawn into the center of the embryo, which enlarges the intercellular spaces, forming this liquid-filled opening. Now, mammal eggs contain very little yolk. They undergo holoblastic cleavage. They form a blastocyst, which is made up of the trophoblast, which is the outer layer of cells, and the blastocele ends up contributing to the placenta. Additionally, the inner cell mass, which is located at one pole of the blastocyst, forms the developing embryo. And birds carry out meroblastic cleavage because the eggs contain a substantial amount of yolk, and the developing embryo is referred to as a blastodisc. And there's a very small blastocele that's formed in the bird egg that is in between the yolk and the blastodisc. So in conclusion today, we looked at sperm penetration and the fusion of the sperm to the cell membrane of the egg, and also egg activation and two types of blockage to polyspermy that take place, and the fusion of the nuclei from the gametes. And additionally, we looked at some of the similarities and differences between types of cleavage in embryos, and looked at holoblastic and meroblastic cleavage, and also compared and contrasted mammal early development and bird.